Okay, so now what's been autosomal linkage? Autosomal linkage, the idea of autosomal linkage here means that these two genes, they are linked at the autosome, means that the chromosome that's nothing to do with the sexual characteristic. Okay, so what actually happened here is one of this person called Thomas Hans Morgans tried to mimic or simulates back this theory of inheritance through the Mendel experiment, but he failed to get that particular ratio. It means that 9331 no longer applicable. Okay, in this case, involve the dihybrid, the modifications on the dihybrid cross. Okay, and we do know that the typical ratio is 9331, but we can't get that particular ratio anymore. Okay, so Morgan also showed that independent assortments does not apply. This is very, very important part. Okay, independent assortments does not apply. So why independent assortments does not apply? Let us look at these conditions now. If I have two genes, they are located on the same chromosome. Let's say, for example, gene A. Gene B. So both of them, they are located on the same chromosome. So it means that no matter how you're going to segregate them, you will always get AB, big A, big B together, small a, small b together. The ratio will be half. Rather than if A and B, they are located on two different chromosomes. So what will happen here will be, if this is big A allele, small a allele. So big B allele, small b allele. So in this case, if I segregate them, okay, then I will be able to get big A, big B together, small a, small b together. But because of independent assortments take place, okay, so arrangements or these alignments of the chromosome during metaphase 2 may not be the same, right? May not be the same as the first one. So you may have big A allele here, small a allele. Here, instead of big B, you have small b and big B. So if the law of segregation applied in this case, I will get another combinations of big A, small a, big B. And these four combinations will be in equal chance. So 1/4, 1/4, 1/4, 1/4. You get the idea? The entire thing here, you can see the two different things because this is the linkage. So autosomal linkage. So it means that no independent assortment that take place between these two genes. But here, because they are not linked, so when they are not linked, so it means that independent assortments take place. Are you clear? Independent assortment take place. If you need to take a photo, so I zoom it in here as okay. Sorry, not zoom in, I zoom out here so that you'll be able to see and compare the difference. So what we're going to discuss now is this autosomal linkage, how this autosomal linkage will change, okay, the Mendel's 9331s when we have the heterozygous cross with the heterozygous, okay? So let us look at the notes now. So the experiment have been done on this, what we call the fruit flies. So they are locus controlling the body color. So we have the dominant allele big B, code for the gray color, and recessive small b, code for the black body. So gray versus black, gray is a dominant trait. Okay, and then another locus, okay, another gene locus control the body, sorry, the wing shapes, my mistake. The shape of the wing. So we have the big V allele, in the exam, don't choose the big V or small v, because sometimes it's quite difficult. When you type it, you can see the big, capital B and capital V and a small letter v. But when you write it, you may tend to write it wrongly. Okay, so don't choose V. V is not a good alphabet to be used when you want to represent the particular allele. Okay, so big V, small v. So big V code for normal wing, which is dominant, and recessive V will code for short wing called vestigial wing. Okay, so if we start with a pure breeding gray body, if I start with pure breeding gray body, normal wing, so means that pure breeding basically means that homozygous. So big B, big B, big V, big B. When you cross with homozygous black body vestigial wing, small b, small v, small b, small b, small v, small v, or F1, no problems, we'll have the gray body and normal wings, right? Okay, but they are heterozygous. So this one's nothing to do. In this case, you can see that it's similar to Mendel's dihybrid cross. Okay, so what we're going to do here is I'm going to start it with F1. Okay, sorry. Assume that, okay, in this case, okay, sorry, a bit laggy here. Now, first thing, if I cross normal, I can slowly follow. Okay, sorry, gray body, normal wing. Okay, gray body, normal wing. So if you cross gray body, normal wing, because first, this is the parental generation. So I put it as a body, big B, big B, big V, big V. Okay, if I cross it with the black body, okay, vestigial wing or short wing. Can I put short wing? Okay, so means that it's a small b, small b, small v, small v. Okay, so what are the gametes? In this case, you look at the gametes genotype. So O will have big B, big V. Correct or not? Here we have O, small b, small v. So when fertilization takes place, what will be my F1? My F1 will show all of them big B, small b, big V, small v. Correct or not? Okay, nothing special here, right? Nothing special. Okay, at this moment, so in this case, all F1 generation will show me with the gray body, normal wing. No issues. But if I cross gray body, normal wing, okay, so big B, small b, big V, small v, with a black body again, short wing, which will have small b, small b, small v, small b. So if according to the dihybrid cross, independent assortments take place, you can see that what is my gametes genotype. You look at my gamete genotypes, I will have big B, big V together. I will have big B, small v together. I'm going to have small b, big V together. And I'm going to have small b, small v together. So in the chance of 1/4 each of them. And for this black body short wing, I'm going to have all of the gametes will have big, small b, small v, small b, small v. So in terms of fertilization take place, what we're going to have for the F2 generations? You're going to have big B, small v, big V, small v, correct or not? So this one will give us the gray body, short wing, 1/4. I'm going to have another 1/4, big B, small v, small v, small v. So this one will give us the gray body. Sorry, this one's great body, normal wing. I write full. Gray body, okay, normal wing. This one will give us a gray body, but short wing. Then if I take this, so we have small b, big V, small v. So this one also 1/4 chance, but this one I'm going to get black body, normal wing. Correct or not? And then if cross the last one, I'm going to have 1/4 small b, small b, small v, small v. I'm going to get black body, short wing. And in terms of ratio, you can see that it's 1 to 1 to 1 to 1. Correct. Okay, look at this. Am I right? You look at this again. I'm going to show you. So please follow. If independent assortment take place. If independent assortment take place. So here with the assumptions that independent assortment take place. So therefore, you will have these 4 types of gametes, right? Independent assortments. So it means that they are not the linked gene. So I will get the ratio 1 to 1 to 1 to 1, or each of them 25%. Okay, but what Morgans have done here, Morgans didn't get this ratio 1 to 1 to 1 to 1. Morgan didn't get. Okay, so what happened here is, if the loci governing the body color and also the wing shape, they are unlinked. So it means that on different chromosome, so it means that independent assortments will allow us to have the heterozygous parent to produce 4 different kind of gametes in equal number or equal chance. So it means that you have the big B, big B, small v, big V, small v, small v. I drawn to you guys, right? For you guys, right? So the cross will give us 25% gray body, normal wings, 25% black body vestigial wings, or we call it as a short wing. 25% we have the gray body, vestigial wing, and 25% we have the black body, normal wing. So it means that in this case will be 1 to 1 to 1 to 1 ratio. Can you see that? So you get 1 to 1 to 1 to 1 ratio. But when Morgans carry out this experiment, I expect to have this. I expect to have this. But when Morgans carry out the experiment, Morgans didn't get this ratio. So what Morgan actually gets? Morgans actually get this. You look at this, expected ratio here is 1 to 1 to 1 to 1, but Morgan's actual result shows a totally different what we call this ratio, because you can see that for the Morgan experiment, gray body normal wing, black body short wing in high number, 965 to 944. So it means that in high number. And then gray vestigial or gray short and black normal in a small number. Can I say that? So this ratio is no longer very, very clear cut. It's no longer 1 to 1 to 1 to 1. So it means something is wrong there. Something is wrong. Why? Because if I follow exactly what Mendel have done, independent assortments have taken place, then I should then get the result that actually out from 1 to 1 to 1 to 1. So Morgan actually figured out, is it possible that these 2 genes, they are located on the same chromosome? So means that A and B, big A, big B, always together, small b, always together. So let us look at if independent assortments does not take place. Okay, so again, so entire story begin again. So if both of them they are linked, then we have to use a special way to write it. Okay, so first I cross my parental generation. Now look at this, parental generation. I started with the gray body, normal wing. Okay, if they are linked, so means that they are going to have big A with big B together, big A, big B together. So if I draw the chromosome out, you will see something like this. Guys, let me move it down a bit. So the chromosome will show us this. Okay, so big A, big A, big B, big B. So they are linked. So means yeah, they are linked. So that's why big A, big B, I write it together. Big A, big B, I put a bracket to show that they are linked. This is how we write it. So if I cross it with black, short wing. So I will have small a, small b, small a, small b. Can I see that? So if I draw the chromosome out, it would be something like this. Small a, small a, small b, small b. So that's why small a, small b together, small a, small b together. So when fertilization takes place, this is the genotype. I'm going to have the gamete genotype. So in terms of gamete genotypes here, in terms of gamete genotypes, then you will realize that you're going to have O, big A, big B, so similar. So that's why the first generation, you don't see any different. So when fertilization takes place, I'm going to have O, big A, big B, small a, small b. So all of them will show us the gray body, normal wing. Okay, gray body, normal wing. So if I take this gray body, normal wing, the F1, right? Okay, I take the F1 gray body, normal wing, which is big A, big B, small a, small b, cannot cross with a black body. Okay, black body, short wing. Small a, small b, small a, small b. What we're going to have here is, this is my gametes genotype. I'm going to see that half of my gametes, the gametes will be big A, big B. Half of them will be small a, small b. Can I see that? Here, all of them will be small a, small b. So fertilization takes place, I will get my F2 generation. My F2 generations will be, if we take this one and this one, I will get big A, big B together, small a, small b together. So the chances is 1/2. So this one will give us the gray body, normal wing. Can I see that? Gray body, normal wing. Another ones, if I take this one and this one, fertilize together, I will have the half of the chance, small a, small b, small a, small b. So this one will give us the black body, short wing. So what is the expected ratio here? 1 to 1. Can I see that? So it means that 50% will give us the gray body, normal wing. Another 50% will give us a black body, short wing. If both of them, they are linked. Okay, if you want to take a photo, you can. Can I see that? If they are linked. So if they are linked, so basically means that we are going to get the ratio of 1 to 1. We're going to get the ratio 1 to 1. So 50%, 50%. So this is means by the sentence here. If autosomal linkage were complete, so means that they're located on the same chromosome, then the mating between the F1 individual, big B, small v, big V, small v, heterozygous, with the homozygous recessive black body, vestigial wing or short wing, we were going to produce 50%, 50%. So 50% will be gray body, normal wing, I show you, right? Then another 50% black body, vestigial wing. So in 1-to-1 ratio. But now, if you look at this data again, not correct, because I still cannot get 1-to-1 ratio. My ratio here is the gray body. This now you can see that gray normal, black vestigial, or black short. So in 1-to-1 ratio, it seems correct, 1-to-1 ratio. But how about this? Where these two come from? Why we have these two phenotype? Because according to my cross, I only will have gray body, normal wing, black body, short wing. So where these two, gray body, short wing, black body, normal wing, and they're always in almost the 1-to-1 ratio as well. Can I see that? But in this case, their 1-to-1 cannot compare with the original 1-to-1. So what happened to this? If you look at this, if you look at this two, gray, I highlight with different color, gray, gray. Can I see that? Black, black. Okay, I use blue color, normal, normal. It's a green color, vestigial, vestigial. Do you realize that both of them mix now? It's a recombinant type, we call it. Recombinant types. It's no longer gray normal or one group, black vestigial or black short one group, but now there is a mix of the allele. So whenever you think about mix of the allele, then think about crossing over. So the crossing over actually gives us a small number of the mix, or what we call the recombinant types of the gametes. Are you clear? So what's mean how this thing will arise, how this thing can happen? Basically, we have to go back to the meiosis again. Okay, so you look at the meiosis. So during the prophase 1 and metaphase 1. Because of prophase 1 and metaphase 1, we do know that homologous chromosome pair, and then they form tetrads, crossing over take place. Now they are located. Okay, where they're located? They're located at the metaphase plate. So this will be something that we can look at it. So, oops, sorry. I think better use this color. Now, chromosomes is something like this. Okay, so later I show crossing over. So I have the big A allele here. This one also, okay. Then I have a big B allele. Those I highlight with the color, okay. And then those I didn't highlight, they are recessive, small a. Those I didn't color, small b. So if crossing over actually take place here at this arm, okay. So when I finish the meiosis 1, we're going to get 2 cells. Each of them will become haploid. Okay, each of them will become haploid. So we're going to get the first chromosome after crossing over. So when after crossing over, you will get this big A. But because of crossing over take place at this arm, you can see that small a. But here I still have the big B, big B. Okay, so this side I will get big A and then small a. Then I'm going to have small b, small b. Are you clear? Okay, then after meiosis 2, I will have 4 gametes, each of them with 1 sister chromatid. But what you're going to see here is big A, small a, big A, small a, big B, this one, big B, small b, small b. And if you compare to the original, before crossing over, big A and big B always together, small a and small b always together. So this kind of gamete we call as a parental gametes, because they resemble back. Can I see that? So parental gamete. Because why? Because they no change. They look like parental, no change at all. But these two, you can see that because of crossing over, we recombine the gametes. We recombine the allele. So when recombine the allele, this is called recombinant. Okay, recombinant gamete. So when fertilization takes place, parental gametes will give us the parental phenotype. Okay, and we cross it with the homozygous recessive. So this one, let's say for example, in this case, it will give us the gray body, normal wing. So gray body, normal wing is a parental phenotype, correct or not? Right? Initially, we cross also gray body, normal wing. This one, this parental, also give us the parental phenotype. So this one will give us the black body, vestigial or short wing. Okay, so these two will in high proportions, means that their number will be high. Because why? No crossing over. When no crossing over, automatically their number will be high. But this small crossing over will give us a small number. These two gametes, okay, will be in small number. Will be in small number. They're going to give us the recombinant phenotype. Because we recombine the allele, right? Okay, in one chromosome. So recombinant phenotype, they're going to give us later. For example, in this case, we have 2 possibilities. Either ones we have the mixture of gray body with short wing. Or another possible phenotypes will be the black body, okay, normal wing, which is a mixture of both parental types. So this one, we name them as the recombinant phenotypes. So in this case, they occur in a small number. Okay, they occur in a small number. Okay, later I will summarize again. So you want to copy it out, you can. Okay, or not copy it out, it means you want to take okay, photo screenshots. Please do it. So this is explanations of the why we have a small number of the recombinant phenotypes is due to the crossing over. Okay, so later I will summarize in the question, exam question, how you answer the question. Okay, how we answer the question, because autosomal linkage is a little bit confusing. I always say that because they have 2 methods to do this the answer. Okay, 2 methods. I mean, we need to use do it. Okay, okay, so now let us go back to the notes and then you'll see the overall picture. So this one just now I have shown to you guys already. See, when we have male flights, which is gray body, normal wing, cross with the gray body. This is not normal wing, it's a bit short wing. My mistake again. Okay, gray body, short wings. Then you will get the gametes genotype. So always remember for gamete genotypes, probability and circle. So in this case, you can see that I'm going to get the gray body, normal wing, black body, vestigial wing, or the short wing in the ratio of 1 to 1. Where if you compare to the result here, you won't get it. Are you clear? You only get here 1 to 1. So we do have the recombinant types. Can I see that? Recombinant type of the gamete or recombinant gametes. So this because of the crossing over. Okay, we have crossing over, so they occur in the small number. So the gray body vestigial wing, okay, gray body vestigial wing, recombinant. Okay, then we have the black body normal wing. So this kind we call them as a recombinant phenotypes. So this will arise because of the crossing over between the gene loci, okay, during the prophase 1. Okay, so these two, number 9, number 10, is not in our syllabus, but question come out before. So I put it in. From the data here, we can actually calculate the percentage of crossing over. How we calculate the percentage of crossing over, or sometimes we also call it a crossing over value. Crossing over value. Now, what's mean a crossing over value? So crossing over value is a percentage. We're going to get the recombinant versus, okay, parental plus recombinant multiplied with 100%. Now, how to get it? If you look at this, recombinant types here will be 206, 185, right? Okay, remove it. Look at this. These two, they are recombinant. These two, they are parental. So recombinant here will be 206 plus 185 divided by the total. Can I see that? The total here, if you calculate, okay, 965 plus 944 plus 206 plus 185 will be 2300 multiplied with 100. You will get about 17%. So this is a crossing over value or percentage of crossing over. So question asked to calculate before. Okay, can I see that? So what does this 17% mean? The larger the value, the percentage crossing over, the COV, when COV is high, means that the distance between two allele or two gene loci further. So means that if I have gene A and gene B, if I draw it, so this is a distance. So distance here we call them as a 17 map unit. They are 17 map units. So if I have the D and E, gene D and gene E, if they say they are the COV, crossing over value equal to, let's say, 10%. So it means that D and E will be shorter. So with the distance of 10 map units, they are arbitrary units. So if let's say here, if I draw it with 17 centimeter, so D and E, I have to draw it as 10 centimeter in terms of the ratio. Because why? Crossing over number lower means that they are nearer. These two genes, they are nearer. Okay, so there is a positive correlation between the frequency of the recombinant of two loci or the crossing over. So therefore, you can convert them into a map unit. Okay, so the higher the value, the longer the distance between that two particular, okay, two particular genes. Okay, gene loci. So with this, I've done for the crossing over. Now these ones are, if you are interested to know more, can always come and find me. This is not in our syllabus, just for your information only. You can try, okay, try to figure it out. Okay, so perhaps when we have free times, then I can go through with you guys. Okay, so these are the question. This another question you can try. It's what is called as the gene maps. So we have A, we have gene locus B, C, D, and E. So how we map them? Is it A, B, C, D, E, or A, B, C, E, D, or A, C, B, D, E? So based on this crossing over value, you will be able to actually figure out, okay, what is the sequence. If let's say I start with A, so where we're going to put a B, C, D, and E based on the crossing over value. You can try it when you're free, but basically this one is not in our syllabus for the gene mapping. Okay, so with this, I have done for the crossing this autosomal linkage. If you have any questions, feel free to ask me. Okay.